Hello, my name is Kaylin Wilkie, and thank you for joining this virtual seminar on designing the perfect PROTAC. I am a product manager in our emerging chemical synthesis team, and together we introduce new products to accelerate drug discovery. This includes everything from lead discovery to the synthesis of small molecules and optimizing those reactions. Today we'll be discussing targeted protein degradation, which is a growing field that uses small molecules to selectively eliminate target proteins from cells. Protein degradation is a process that happens in our cells all the time. These E3 ubiquitin ligases, shown here in purple, seek out and tag old, damaged, or misfolded proteins and label them with ubiquitin. This flags those proteins for degradation down the proteasome or the cellular trash can. Chemical biologists discovered they could hijack this process and degrade target proteins. And as many of you know, they do this with small molecules called protein degraders, commonly called protein degraders. These are heterobifunctional molecules that contain a target warhead on one side and E3 ligase ligand on the other, connected by a cross-linker in the middle. When you add these to the biological system, it anchors that target protein to the E3 ligase, allowing for the native cellular machinery to take over. Ubiquitin is added to that target protein and is degraded down the proteasome. So when we think about how degraders are designed, it oftentimes starts with a warhead or inhibitor that has already been identified or exists for a target protein. So it could come from a screening campaign, or maybe it's repurposed from existing drugs. And then you add on a crosslinker and an E3 ligase ligand. It's through this process that we've really come to appreciate some of the advantages of degraders over traditional occupancy-based small molecule inhibition. For example, low affinity target warheads or inhibitors can lead to effective degraders. You can take a promiscuous inhibitor and turn it into a selective degrader. You can use lower concentration of degraders because they work catalytically, meaning that one degrader will degrade one protein and then a next and a next and a next. Another big advantage is the expanded target scope to undruggable proteins. So inhibitors are limited to about 20% of the proteome that have accessible deep binding pockets for receptor binding or enzyme activity, where the inhibitor goes in there, blocks the function and deactivates those proteins. Degraders, on the other hand, can bind anywhere on that target protein as long as it can drag it to the E3 ligase, meaning this is expanding the scope of what we can drug in comparison to traditional inhibitors. Lastly, the protein degraders have been called the chemical equivalent to CRISPR because we see phenotypes similar to gene editing, except with small molecule control. And so for all of these reasons, protein degraders have been identified as a new modality in drug discovery. But their design is not trivial, and this is because you are trying to design one molecule, such as this green PROTAC here, to bind two proteins simultaneously and create this protein-protein interface that may not exist naturally. And so there's different approaches to doing this. One is in very modular fashion, shown here on the right, where you take these four variables and you strategically vary them to create a starting potential degrader library. So that E3 ligase ligand, the exit vector, linker, and target warhead. And then you screen all of these potential degraders to see if any of them work. And so at this point, degrader design is still very empirical. The degraders themselves are multi-step synthesis, and so generating libraries of them is quite a daunting task. So we in chemistry thought about how we can help accelerate researchers' one target warhead to a starting library and some degrader hits. And so we developed and launched a library of degrader building blocks that simplify not only the synthesis of one protein degrader, but a whole library. And so an example of a degrader building block is shown here, where it contains three components. One is an E3 ligase binder, and so we've begun with the most validated ligands for VHL and cereblon. The second component is a linker, and we started with short aliphatic chains and flexible pegylated linkers. We also have just recently launched some mixed linkers that contain both hydrophobic and hydrophilic properties. And then on the end of that linker is what we call the terminal chemistry, or the functional group that a researcher would use to add his or her target warhead. And so this is where the library generation is really streamlined, because if you pick one of these reactive groups, for example, the amine, this is a slide showing all of our amino-terminated degrader building blocks. You can start with one target warhead and quickly generate, in parallel fashion, a starting library of protein degraders to screen for degradation of your target in cells. And so to acquire these degrader building blocks, the fastest way is to go through our stocked catalog pre-pack. We have over 80 of these available at 50 milligram quantities. And if there's something that's obviously missing, or something you know you will need in the future or on a routine basis, please let us know. You can also access all of these at quantities greater than 50 milligrams. So we're experienced at providing these at bulk scale. And we also have a custom synthesis team that can provide custom building blocks or full degraders that are specific to your research. So you can reach out to us about any of these methods. To access this portfolio, you can follow this link here, and it will bring you to our portal. A couple things to note. One, you can click on this link to access all of our degrader building blocks. Every time there's a new launch, it will get added here. We have a new review in the Ultra Chimica Acta that discusses the development of small molecule degraders. We have a listing of related products, and one of our newest launches is the DTAG 13 probe, which is extremely helpful for protein knockdown experiments in biology. And lastly, if you will be at the TPD Summit in Boston in October, organized by Hanson Wade, we will be there and would love to talk to you. So one of the questions we get commonly is what's next, and we know there's a wish list for the Protacs toolbox. So we're really excited about some of the launches coming out later this year. You'll see degrader building blocks that include new E3 ligands, new exit vectors, some diversity and rigidity in that linker region, as well as some photo-switchable derivatives. If you're designing Protacs or protein degraders for undruggable targets, you may also be interested in some of our new products coming out later in 2020 for lead discovery, ligandability, or protein-protein interactions. And I think the collective question for you, me, and the field is really what will degrade next and how we'll do it. And so as we continue to see new methods and flavors of degraders emerge, and as you think about your future programs in protein degradation, if we can be of help or you're interested in ideating with us, please reach out. So thank you for your time. Best of luck in your degrader research, and have a good day.